The goal of the Myeloid Network is to bring us together, the international community of people who study myeloid cells in cancer, infectious diseases, inflammation, and others, to exchange ideas, give talks, support trainees, and educational efforts. We have a website that Jennifer has designed where we hope to be able to invite you to advertise for postdocs, submit questions, and submit ideas for what we can do as a group of international researchers. And we hope you'll continue to join us, and we will have monthly seminars, and I will begin the first seminar. So now I'll turn it over to Jennifer.
Great. Thank you, Dr. Varner. I'm so excited for the seminar series. So thank you for all your work too, and also to Tyler from your team for helping with putting all this together. So I want to introduce Dr. Varner, our first speaker in the Myeloid Network seminar series. Dr. Varner is a professor in the Department of Pathology and Medicine at the University of California, San Diego. She's the Director of Faculty Mentoring for the Department of Pathology, and she's the co-leader of the Solid Tumor Therapeutic Program at the Moores UCSD Cancer Center. As many of you know, her work has focused on the mechanisms regulating the tumor microenvironment and inflammation in cancer, and she's identified novel mechanisms by which macrophages promote tumor growth and has developed a number of approaches to treat cancer patients by targeting tumor macrophage signal transduction, including a focus on PI3 kinase gamma. Novel therapeutics target against PI3 kinase gamma have been developed together with Infinity Pharmaceuticals and the Varner Lab, and they're currently in testing in 5 different phase 2 clinical trials. Dr. Varner is a recipient of many awards, including the V Foundation Award for Translational Medicine, the AACR Landon Foundation Innovator Award for International Collaboration, and the Lustgarten Foundation Innovator Award for Pancreatic Cancer for her work on immune oncology therapeutics. So today she's going to present her timely work on macrophages and COVID-19. I'll ask you to stay muted throughout the talk, but and I can collect questions from the chat, so please enter them into the chat. Following the 45-minute presentation plus the 15 minutes of Q&A, Dr. Varner has agreed to stay on for longer to answer any additional questions. We hope that this will garner some more intimate discussion and that folks can turn on their camera and ask their questions directly. So please stay on after the hour seminar if you can. So Dr. Varner, I'll turn it back over to you.
Okay. All right. It gives me great pleasure to speak to you today about our work targeting macrophage trafficking in COVID-19 disease. So we all know macrophages are associated with infectious diseases and tumor progression, and that they play important roles in homeostasis, in recognizing pathogens and eliminating them. They also play roles in embryonic development, including vascular development in the neonatal eye. And unfortunately, they also contribute to tumor progression and metastasis, as we can see here in this pancreatic ductal adenocarcinoma. So normally, macrophages should play important roles in innate immunity, but also in collaborating with B cells and T cells to activate them and control their function. However, in cancer, these processes can go awry. And, but also, as we've seen in COVID-19 and other similar diseases, macrophages can also contribute to pathogenesis. So over 10 years ago, Michael Schmidt and my lab decided to focus on understanding the mechanisms regulating myeloid cell recruitment to tissues. At the time, it was known that several chemokines, such as SDF1 and CCL2, and also cytokines, such as IL-1 or VEGF, could play roles in recruiting myeloid cells into tumors, where they could then contribute to tumor progression as macrophages or granulocytes. So we were studying integrin biology at the time, and we asked which integrins were important for allowing this process to occur. So you may know integrins are heterodimeric cell adhesion proteins that are expressed abundantly on the surface of leukocytes, where they typically are in an inactive folded state, and they're unable to attach to facilitate attachment of cells to endothelial cells until they're activated by a signal such as a chemokine or a cytokine. Once they are activated, those integrins unfold and allow attachment to the endothelium and extravasation into tissues. So we wanted to know which integrins play a role in this process, and Michael undertook a series of assays to determine that. So when integrins are inactive, they're folded and they're unable to mediate attachment. When they're incubated with chemokines or cytokines, they unfold and they mediate attachment. So he devised an attachment assay and showed that a variety of different factors could all stimulate increased attachment of circulating monocytes and granulocytes to endothelial cell monolayers in vitro. The only integrin that seemed to be responsible for that event turned out to be integrin alpha 4 beta 1, because antibody and other inhibitor blockers of this process could completely suppress this activation event, while inhibitors of the common integrin C11b or alpha M had no effect, and other integrins also had no effect. But we also noted that there were multiple cytokines and chemokines that could activate this integrin, so we speculated there would be a common signaling pathway behind that. So we sought a variety of inhibitors of signal transduction processes and found surprisingly that only one signaling agent appeared to control this process, and that was the isoform of PI3 kinase gamma. So inhibitors or SIR knockdown or knockouts of this kinase were completely able to suppress this attachment event. And so that led Michael through a series of papers, as well as additional postdocs who followed him, to decipher this pathway. We realized that different receptors, such as receptor tyrosine kinases, G protein coupled receptors, and interleukin receptors could activate PI3 kinase gamma by way of RAS. And that led to the signaling cascade that facilitated attachment of the integrin to the actin cytoskeleton by way of myosin 2. This then allows cells to extravasate and participate in angiogenesis, tumor immune suppression, and other functions in the tumor microenvironment. But this process also allows cells to extravasate during inflammatory processes, such as in COVID-19. So PI3 kinase gamma is a very unique class 1 PI3 kinase isoform. It has a unique N-terminus where it lacks the P85 binding domain found in the other class 1 isoforms, and it is taken to the membrane by two distinct regulatory subunits, which are different from those for the class 1A subunits. And then it's also unique in that it's the major PI3 kinase isoform in myeloid cells. It's much more rarely expressed in lymphocytes and seldomly expressed in epithelial cells or tumor cells, except under certain conditions. The structure's been crystallized for quite a while, and a number of inhibitors have been made and been in the clinic, but the most selective such inhibitor is now in the clinic, and this is eganelisib, or also called IPI549, a highly active, highly selective inhibitor that we have used in combination with Infinity Pharmaceuticals to evaluate the effect on cancer in animals. And so Megan and Michael Schmidt both showed through a series of animal models that inhibiting PI3 kinase gamma could substantially suppress inflammation in the tumor microenvironment, either implanted tumors or genetically engineered tumors, such as this genetically engineered breast carcinoma, and that inhibiting PI3 kinase gamma can block tumor growth to varying degrees depending upon the tumor model. And in fact, this IPI549 inhibitor was very active in suppressing tumor growth in a similar manner as the knockout mouse. So the other function that we realized PI3 kinase gamma did was that it repolarizes macrophages in the tumor microenvironment. When we looked at the gene expression content of knockout or drug-treated tumors, we find that there's a substantial change that suggests an activation of antigen presentation, innate immunity, and T cell activation. And when we look at tumor-associated macrophages isolated from these tumors, those would be residual macrophages that were not inhibited from trafficking into the tumor. We find an upregulation of a variety of innate immune factors. Importantly, we see a strong upregulation of MHC class II expression on the surface of macrophages in breast and lung tumors, and so far in all tumors that we've studied. So this led us to ask whether these were the consequence of inhibiting PI3 kinase gamma in myeloid cells trafficking to the tumor by reducing the tumor immune suppressive content, or was this actually a function of PI3 kinase gamma on myeloid cells directly? So we looked at in vitro models of macrophage polarization, where we treated macrophages with either interferon gamma plus LPS to induce a pro-inflammatory response, or IL-4 to induce a more wound healing alternatively activated response. And then we looked at the fold change of various genes in response to these versus wild type in response to these factors. So what we found is there's a constitutive upregulation of MHC associated molecules in these macrophages. So both MHC minor and major molecules, including the regulator of the MHC family, C2TA, and the regulatory subunit, CD74, are all upregulated. There's upregulation of a variety of pro-inflammatory and immune stimulatory factors, depending on the degree depends upon the stimulus. But importantly, there's a strong downregulation of chemokines and cytokines, as well as other factors that are associated with myeloid cell recruitment and immune suppression. So when we looked in vitro at specific genes, what we could see is regardless of the stimulus, we saw a strong upregulation of factors that could assist in the activation of T cells, particularly IL-12 is strongly upregulated. And we also see downregulation of immune suppressive factors like IL-10, TGF-beta arginase, and CCL2, which is important in recruiting myeloid cells into tumors. We also, in vitro, we see a strong production of IL-12 protein, thousands-fold increase in IL-12 expression and a strong decrease in IL-10. These suggest then that PI3 kinase gamma inhibition could provoke a positive immune response in tissues. So the pathways that are activated are all immune response pathways, regulation of T cell activation, and so on. And so Megan then found that the basic cause of this is the increase in NF-kappa B activation in PI3 kinase gamma knockout cells. There's strong upregulation and sustained activation of NF-kappa B. And we've seen that recently in a TechSeq data in collaboration with our colleague at the Salk Institute, Diana Hargraves, and her graduate student, Jingwen Lao. So essentially what we find is that PI3 kinase gamma is an inhibitor of NF-kappa B that probably gets activated during toll-like receptor activation, and is sort of a feedback inhibitor. While if you inhibit PI3 kinase gamma, you can sustain NF-kappa B activation, leading to beneficial effects on the tumor microenvironment. And then that leads to an upregulated accumulation of CD8 positive T cells, as shown by Hideyuki Takahashi, who's now assistant professor in Japan, and a strong upregulation of activated effector cells in the tumor microenvironment. And that's demonstrated by increased granzyme B expression. So Infinity Pharmaceuticals has developed this first-in-class oral selectively inhibitor of PI3 kinase gamma that's currently has been in 5 clinical trials, and is currently entering a phase 3 trial for registration purposes, and it's showing positive effects in a number of different kinds of tumors. So when the pandemic hit, we thought, and it became clear that there was a problem with inflammation in patients who died from COVID. We decided to collaborate with Infinity Pharmaceuticals to investigate whether PI3 kinase gamma inhibition could be useful as a therapeutic approach for SARS-CoV-2 related acute respiratory distress syndrome. So the concept of respiratory distress syndrome in many diseases is that in response to a pathogen or injury to the lung, alveolar macrophages recruit T cells that can then, and damaged epithelium recruit cells that lead to the production of cytokines that recruit monocytes and granulocytes into the lung. And if the damage is severe, the recruitment becomes substantial and can also lead to chronic recruitment of further cells by the myeloid cells themselves. And then this leads to fibrosis and filling of the lungs with damaging cells. So our pathologists here at UCSD have evaluated tissues from COVID patients, and what you can see in the lungs of these patients is a number of factors. You can see in these different images that there are too many macrophages present within the alveolar spaces. Sometimes the alveolar spaces are completely full of cells, and the blood vessels are congested, as you can see here by the red staining. So this is an abnormal appearance for a lung. While an alveolar space should be clear and free, and the vessels should be much less expanded and obvious. So in our lab, we were able to access tissue from normal lung and also from COVID-19 patients who had passed away at UCSD hospitals. And we got tissue from our collaborators in pathology and medicine, Jingjing Hu, Hanaruso Omo, Asa Gaboni, Laura Crotty-Alexander, Li Zhu, Xinzan, and Sumita Das, who've been great collaborators with us during these difficult times. And Angus Mgraber-Medin is a postdoc in my lab who worked out the staining conditions to be able to demonstrate that in normal lung, clearly you have a few resident macrophages, not too many. While in the COVID lung, you can see the dramatic scenario of far too many macrophages, sometimes completely filling the alveolar spaces. So there's about a 500-fold increase in the number of macrophages in tissues. He also observed an increase in granulocytes present within tissues by staining for myeloperoxidase. So in normal lung, again, relatively few, but in the COVID lung, you can see the tissue is full of these. The neutrophil content is a little more variable than the macrophage content. So then we turn to our good collaborator, Lisa Cousins, and her postdoc, Courtney Betts, and their multiplex immunohistochemistry team, who performed multiplex immunohistochemistry on these same tissues that we had accessed from UCSD. So what multiplex IHC is in their lab is incubation with primary antibodies detection, scanning of the slide, then removing the chromogen and the antibody and repeating cycles. And with a series of slides, then you can identify immune cell content using a variety of different antibodies by co-registering the information and then analyzing. And so using this panel of antibodies that they have well developed and published upon, you can identify a variety of different cell types. So when applied to our lung cancers, our lung specimens, which are 15 COVID-19 mortality cases and associated normal lung from stage 1 lung cancer patients undergoing surgery. What was really clear is there's a strong increase in the number of CD11B positive cells seen here in these images, while no particular increase in CD20 or CD3 indicating T cells. And interestingly, there is somewhat of a decrease in the epithelial cell content, as I'll show you in a minute. Importantly, what you see is a strong increase in total macrophages and monocytes, as well as other myeloid cells that can be other macrophages that don't fit quite the definition that I showed you in the previous slide, and then other immune cells. So there is clearly an inflammation response going on in these tissues. Now, if you look at macrophages alone, you can see that there's a very strong increase in the number of macrophages, and especially in CD163 positive macrophages, which is indicative of an M2 type wound healing response seen here. Interestingly, there's no particular change in the content of T cells in these patients, suggesting they really were not responding to the virus disease by mounting a good T cell response. And then in looking at the epithelium, you could see some variability in the presence of epithelial markers, but it does look as if there is a reduction in the total amount of epithelium. And so that is consistent with the destruction of the lung by this virus. Now, to get more information, we were unable to work with tissue that had not been fixed at UCSD, so we turned to a company in Carlsbad, California called BioSpider that had developed a very new and exciting technology to amplify DNA sequences or RNA sequences from tissues and using very small amounts of material. Using this procedure, they use specific oligonucleotides to amplify short sections of genes and then sequence and identify genes based on these tags. And so doing this, we were able then to show, in collaboration with our bioinformatics colleagues, Roman Susick and Katie Fish at UCSD, that the normal lung is substantially different from COVID virus, so from patients who had died from COVID. And interestingly, we found that only a few of the patients actually still showed signs of virus, while most of them had long since cleared the virus and were suffering from the effects of the inflammation and fibrosis. So there are a significantly large number of genes that are downregulated in the COVID positive patients, which are associated with a decreased endothelial cell health. And then there were relatively few chemokines that were upregulated in the COVID patients, suggesting also that the immune response was somewhat awry as well. So when we look at the pathways that were implicated in these sequences, we found that there is a picture of tissue in distress. So there's an overall signature of glycolysis and of unfolded protein responses. EMT transitions suggesting movement toward wound healing and fibrosis. And as we've heard in the news and in the literature, there is an increase in coagulation signatures. Now, it's very surprising, but there is a decrease in your classic inflammatory pathways, and particularly in those that lead to T cell activation. So it's interesting that in these patients whose lungs are full of macrophages, there's clearly signs of a wound healing type M2 type macrophage response. So if we look at the COVID positive patient samples, they are different from the COVID, so those that have active virus. These are these patients who passed away from COVID and those that had active virus infection at the time of their death compared to those that did not, there is a signs of an antiviral immune response because there are genes such as these IFITs, guanylate binding proteins, ISGs, and chemokines that are all suggest that there is an attempt at a virus response. However, in those who did not have this signature, there's no such signature. There's no such antiviral response. So Roman applied a deconvolution strategy to try to understand what the signatures told us about cells that were present in the lungs. And we had isolated bronchial alveolar lavage fluid from patients, and we analyzed the cell types there. So normally in the bronchial alveolar lavage, the cells present would be 85% alveolar macrophages and relatively few neutrophils. But in these patients, it's almost 65% neutrophils and only 35% macrophages. So clearly there is a shift in the natural cells that can be acquired from BALF. And then those are characterized as N2 neutrophils, which have been shown to be wound healing type neutrophils, which promote tumor progression. If we look at the if we look at the lung itself and we compare normal lung to COVID lung, some of the things that stand out in this deconvolution strategy is that there's a variable increase in macrophages in the different patient samples, but there's a strong increase in monocytes. And most importantly, a very strong decrease in alveolar type 2 cells, indicating destruction of the lung. And Roman applied this metric to try to demonstrate the changes. So what you clearly see are increased monocytes and interestingly, goblet cells, which is associated with COPD as well, and a slight increase in mesothelial cells, and then the strong decrease in alveolar type 2 cells. So we wanted to know whether we could potentially apply PI3 kinase gamma inhibition to suppressing this deadly macrophage and neutrophil accumulation in these patients. And so we first checked to see whether PI3 kinase gamma was expressed in macrophages in these patients. And what you can see in these serial section images is that in fact PI3 kinase gamma is expressed in CD68 positive cells present in COVID lungs. And so then we wanted to know, does inhibiting PI3 kinase gamma block recruitment of myeloid cells into tissues in response to inflammatory mediators? So here we injected mice with thioglycolate, which induces a mild inflammatory effect and leads to the presence of numerous bone marrow-derived macrophages present in the peritoneum of animals after 3 days of treatment. So we looked at wild type versus knockout mice, and then also vehicle versus IPI 549 treated mice. And in these images, you can see that in wild type mice, you see a strong accumulation of macrophages present within the peritoneum of these mice. And that's substantially reduced in PI3 kinase gamma knockout mice. And it's also strongly reduced in IPI 549 treated mice seen here. So we tried at the time to use sterile models of inflammation to try to find out what would happen if we induced inflammation in the lungs of animals. So Ryan Shepard, Angus M. Gebremedin, and Mark Paradise in my lab all worked together during our massive shutdown at UCSD to set up these models and develop these strategies. So what we did was we injected mice with LPS acutely, so a one-time injection, or chronically with a lower dose. So we injected daily for several days. Or we injected them with poly-IC, either systemically or intranasally, daily for several days to look at the effect on inflammation and the effect of PI3 kinase gamma inhibition. So some of the measures we looked at was just inflammation, tissue pathology, and a vascular leak. So if you look at normal lungs, you see they have wide open alveoli. You can see red blood cells present within the blood vessels of these alveoli, and then you can, and also the larger vessels here, but you notice that the alveoli are very clear. In the acute LPS treated animals, what you can see is substantial vascular leak because you see red blood cells present within the alveoli. You also see thickening of these septa, which is due to inflammation and also engorgement with blood. And that's also seen here in these vehicle treated animals. But what you see in the PI3 kinase gamma knockout in IPI 549 treated animals is substantially normal lungs. So there's very little of this vascular leak and septal thickening. If we look at the chronic LPS treated animals, we see the damage is even greater. So much more substantial disruption of the normal architecture, lots of vascular leak. And then it's much more normal in the PI3 kinase gamma knockout in IPI 549 treated animals. And that's also true with chronic poly-IC injection. In fact, poly-IC chronically injected does more damage to the lungs than LPS does. And it's substantially protective to inhibit PI3 kinase gamma. Another measure of lung destruction is vascular leak. And so we performed the same studies and injected animals with Evans blue dye shortly before ending the experiment, and then we can remove the lungs and extract the dye and then measure spectrophotometrically to determine the degree of vascular leak. So a lung that has a lot of vascular leak will look blue, and you might even have big pockets of blue here and will yield a darker signal. So when we did that using chronic LPS stimulation, we could see a substantial increase in vascular leak as detected by Evans blue in the wild type animals, and it's strongly reduced when you inhibit PI3 kinase gamma. In fact, it's fairly normal. And the same is true when you inhibit with IPI 549. We also see if we administer poly-IC that we saw a strong reduction in vascular leak in the lung and the heart and the kidney. So clearly IPI 549 and PI3 kinase gamma inhibition through the knockout is protective in these settings. So we wanted to know if this is related to reducing inflammation. So what we did was perform bronchoalveolar lavage on these animals. That's where we flush the lungs with the saline solution, then we spin the cells down in a cytospin and then analyze by Giemsa staining. So what you can see here is that there are substantially fewer macrophages. These are macrophages in the IPI 549 treated animals. And that's quantified here. So there's a strong upregulation of macrophages when you induce this inflammation with LPS, and it's basically brought back down to normal when you inhibit with IPI 549. And then also if you knock out PI3 kinase gamma. We also saw the same thing with chronic LPS. There's even more inflammation in the lungs, and that's substantially reduced by inhibiting with IPI 549 or the knockout. And then finally, the same thing is true with poly-IC. So these models do suggest they're inducing inflammation that leads to recruitment of myeloid cells into the lungs, and particularly into the alveolar spaces, and that inhibiting PI3 kinase gamma is protective. But, macrophages in alveolar spaces are not the only macrophages in lungs. There are also interstitial macrophages. And so we did immunostaining to try to quantify macrophages. And these studies are still ongoing. But what we can see is there are more macrophages associated with the actual lung, the lung tissue, not just in alveolar spaces, and also proximal to blood vessels and to bronchi bronchioles, but there are far fewer in the PI3 kinase gamma knockout and IPI 549 treated animals than in the vehicle and wild type animals, and that's been quantified here. So, so what we find is that lethal COVID is associated with immunosuppressive macrophage and neutrophil recruitment and loss of alveolar function, and that PI3 kinase gamma can promote trafficking of myeloid cells during inflammation. Blocking PI3 kinase gamma suppresses myeloid cell recruitment and can stimulate T cell activation in our studies in tumors. And so we suspect that by blocking PI3 kinase gamma in COVID, we could reduce myeloid cell recruitment and potentially help facilitate a better T cell response to the virus. And that blocking PI3 kinase gamma also prevents vascular leak, which is a major part of this COVID lung destruction. So future directions are that we're currently doing murine coronavirus infection studies here at UCSD, and have arranged with NIH at NIAID to do murine and hamster COVID infection studies. And also studies are ongoing at Tufts with the hamster model. And together with Infinity Pharmaceuticals, we helped to arrange a future clinical trial of this drug in COVID, pending the outcome of these animal studies. So in conclusion, we've shown that PI3 kinase gamma inhibition can be potentially it is helpful in cancer, but it could also be helpful as a therapeutic for SARS-CoV-2 or similar diseases such as fatal inflammation that's associated with flu in elderly patients. So early stage disease, as we know, in young people has been fairly successful in being managed by the immune system, but in elderly people or people who are otherwise infirm, we can see that the immune system goes awry and there's too much inflammation and myeloid cell recruitment, and that inhibiting PI3 kinase gamma may be helpful with that response. So I'd like to thank Angus Gebremedin and Ryan Shepard, who did the lion's share of this work with Mark Paradise, as well as other members of my lab who've been assisting them, and Michael Schmidt, Megan Kaneda, and Hide Takahashi, who did all the work on cancer preceding them. My collaborators at UCSD who have contributed to this work, I've mentioned, Infinity Pharmaceuticals colleagues, Brenda O'Connell, Rosita Algova, Theo Martineau, Larry Block, and Jeff Kutak have all been very helpful and very good collaborators throughout this pandemic. And of course, my collaboration with Lisa Cousins in her lab has always been a pleasure, and my funding is here. So I'd like to thank you all for coming to the Myeloid Network, and to remind you what the website address is, and let us know what you think about this network.
Thank you. Thank you so much, Dr. Varner. That was so great. We have a number of questions, lots of enthusiasm from the group. And I mean, this has just been such a great turnout for the first seminar. We have more than 150 participants. There was no technical glitches. I think this has been such a great success so far. Okay, so for questions that are coming in from the group, Tatiana Novobrantseva asks, which function of the PI3 kinase gamma inhibition is having the bigger effect in acute and chronic inflammation, prevention of myeloid cell recruitment, or macrophage repolarization? And where do you see the use of PI3 kinase in COVID patient clinical manifestation?
Yeah, those are great questions. At the moment, we think it is on macrophage recruitment, but we are investigating polarization. It's a complicated event because polarization in response to PI3 kinase gamma can lead to a strong pro-inflammatory effect. So when we look at acute treatment with PI3 kinase gamma inhibitors, we see we can't see an upregulation of things that you might be worried about, like IL-6 and TNF. But if we look chronically, we see a downregulation. So that to me suggests the effect really is mainly on reducing the content of macrophages with some added benefits from the repolarization, but we mainly have so far focused on characterizing the reduction of recruitment. And as far as the clinical trials go, yeah, we think that the right time to use this would be upon hospitalization. It may be, as long as patients are not coming to the hospital too late, that would be the time when intervention would be feasible and has been shown to be successful. But maybe studies will show in the long run that at some point prior to hospitalization would be the ideal time, but at this point that's not practical.
Matthew Parks says, thanks for your talk. He's wondering how you distinguish the effects of PI3 kinase inhibition on tissue-resident macrophages versus DC in the lung niche.
Mm-hmm. That is something we're looking at in cancer. One of the things that we know from our cancer studies is that inhibiting PI3 kinase gamma does not block the expansion of tissue-resident macrophages in cancer. So since they're not being recruited by active adhesion mechanisms, it doesn't affect their recruitment, but it does affect their polarity. I think that what the deconvolution data that Rome and Sosik did suggest that the large increase is due to monocyte-derived macrophages. It can be really difficult to distinguish the signature of recently recruited macrophages and monocytes because they have very similar properties. And so I think what it's showing is that most of the increase is due to recruited macrophages, and that's where we can have the biggest effect on numbers. However, those recruited macrophages do get into the alveolar spaces.
Yeah, so there's a couple other questions about that, but Matthew also asked if perhaps you're modulating the phenotypic outcome of dendritic cells as well. Do you have any evidence for DC activation?
Yeah, so there's a slight increase in the number of dendritic cells in the COVID patients. What we have in the past looked at dendritic cells in cancer, and we see there's a strong increase in activation of those, potentially even of recruitment. And I would predict that that's what we would see here. We have not yet analyzed that, and that's a great thing for us to look at. Unfortunately, due to the pandemic, our ability to do FACS, which is what you would want to do in this sort of setting, has been severely restricted. I know we've all had problems getting things done during COVID, and that's our biggest problem right now. But that's a great thing, and I think we'll go do that now. Thank you for that suggestion.
Yeah, hopefully this will be the strength of this group, that we'll be able to collaborate together and get more science done with labs that have different access to different technologies. So a question about the repolarization and trafficking. You mentioned that the trafficking and recruitment is probably the major thing that needs to be blocked. So have you thought about other strategies then to block their trafficking?
Well, Stanley Perlman at, I think he's in St. Louis, is a big expert on viruses, and he published a beautiful paper on SARS, where he showed that in mouse models of SARS, the fatal disease was caused by a late inflammation stage caused by a defective interferon response, and that it was largely driven by macrophage recruitment. And he showed that blocking CCR2 was effective in that strategy. So yeah, I think that, and we all know that CCR2 positive monocytes lead to recruited macrophages into tumors. And so I think that CCR2 inhibition is being evaluated. And that's the only strategy I know that's so specific for recruited macrophages so far.
Shanti Ganesh asked, he says, great talk. And they ask, do you think that STAT3 will be expressed by these suppressive macrophages in the lungs of COVID patients? And if so, do you think inhibiting STAT3 would give you similar effects from the PI3 kinase gamma inhibitor?
So maybe. So, you know, it depends on if the macrophages are truly M2-like, then yes, STAT3 would be a big player in that. But if they are not, then maybe not. So, you know, M1 type macrophages don't really use STAT3, they use STAT1. And so I think it depends on if they really are wound healing type. And then yes, I would think it would alter what they do. Wouldn't alter their presence, but it would alter what they do. So it could be interesting.
Nan Zhang asks, they say thanks for the nice talk. Would PI3 kinase gamma inhibition cause accumulation of myeloid cells in the blood, which may potentially cause intravascular coagulation, or would it completely inhibit activation of leukocytes to promote coagulation?
So, you know, studies in animals have not shown that there's any particular increase in cells in the blood. You know, that would be the number of cells trafficking in your blood vessels is so much larger than what gets into your tissues that I think it's a drop in the bucket. So that it doesn't substantially lead to an increase in cells in the blood. But what was the other question?
I guess they're just asking about intravascular coagulation.
We've actually been looking at whether or not inhibiting PI3 kinase gamma could be beneficial in that case. So inhibiting PI3 kinase gamma changes quite a few things, including upregulating TPA. So TPA could be beneficial because it is the clot buster. And so if macrophages do play a role in regulating clot dissolution. So it's possible it could be beneficial. We've been trying to get studies like that done, and I think that's a very important aspect. You know, if the clotting is due to destruction of the endothelium by the virus, then I'm not sure this will help, except that it may be helpful in breaking up those clots. So I think we have to see, but it's a great question.
Federico Verga, they also say very nice talk. They were wondering, since in the lethal phase, the macrophages show immunosuppressive phenotype, what do you think they're doing in the late phase?
I think they're promoting wound healing. So the other thing you see if you look at these lungs is they are very fibrotic. Many of them are showing signs of wound healing, very much like a injured lungs from COPD or like bleomycin treated lungs from animals. And so I think that they are promoting wound healing, which is unfortunately what we've shown leads to more fibrosis. So it's an attempt to repair the damage done to the lung epithelium by laying down new tissue, which is scar tissue. And that also is most likely what's causing the inability of people to recover lung function. It's too much scar tissue. Yeah. So I think,
let's see.
Yeah, so we got to prevent this destruction. You know, if the big question still remains is the destruction by the virus too much for anything to repair, you know? But it does seem as if treating patients who recently hospitalized with glucocorticoids and remdesivir and other assisted therapy can help them recover. So that suggests that if you intervene at the right time, you can promote lung recovery. And
you're suggesting it's early that we need to intervene?
Yeah, as soon as somebody feels like they have to go to the hospital. Yeah.
This is a question about side effects. Do you note any, does your PI3 kinase inhibitor have any side effects in your mouse model, and any differences in mortality among LPS treated groups?
Actually, there's increased survival in LPS treated animals, substantially increased, and in animals with tumors, a substantially increased survival, and even in some of the clinical trials, there's substantially increased survival of patients who are treated with the appropriate dose. Like with any drug trial, there is, they have found that certain doses of drug in combination with other drugs can lead to side effects. So provided the dosing is optimized, there are no substantial adverse effects. So, in the knockout mouse, when first characterized, researchers discovered there was an increase in skin infections due to the inhibition of granulocytes trafficking. So there's always that potential, but they haven't seen during clinical trials any particular such effects. I think that I would predict nothing significant. And in fact, that's probably because it's not a very widely expressed molecule. It's not on all of your epithelial cells or even on all of your leukocytes. So that makes it a better target.
So actually related to that, there's a question about PI3 kinase gamma on endothelial cells.
It is, interestingly. And so some of the vascular leak effects are probably direct. So it has been shown by the lab of David Cherish at UCSD in the past that it can regulate vascular leak. And we are looking now directly to determine whether we can reproduce that and to separate those using various animal models. But that it could be a beneficial effect of this drug, whereas we're targeting macrophages, but we may also be targeting the endothelium and preventing the endothelial cells, cell-to-cell junctions from separating, which is what is the cause of vascular leak. Of course, if vascular leak is due to destruction of the endothelium, apoptosis of endothelial cells, then we don't know what effect that would have. But if it's due to this relaxing of the cell-to-cell junctions, then definitely that's what this can affect.
That was a question from Adam Wright, and he has a second question that's maybe more of a discussion, but among the participants here. But he asks if there's a consensus among the myeloid network on nomenclature used to describe recently emigrated monocytes from the blood to tissue. So when they don't look like macrophages, it's sort of a premature label. Are they now called lung monocytes, which may have various phenotypes, maybe yet to be defined?
I think it's a great question, one that I'm asking. And so I think that would be the perfect question for us all to address here. Maybe we can have a free-flowing discussion in a few minutes about that.
Yeah, so there's just one more question from Abigail Elliott. So they ask, in either the cancer models or the inflammatory models, did you try treating with IPI 549 following the onset of inflammation, i.e. when there's already a large number of macrophages recruited? And how long would you have to treat in order to substantially decrease macrophages at the site of inflammation?
Yeah, that's a great question. I have to say we haven't done that yet, but we're about to do that in animal models of virus infection. Obviously, that's an important question to ask. And there are models of the short-term models of inflammation are short. So in order to make a difference, we treated from the beginning. But with the virus inflammation, that should give us some of those answers. Now, with cancer, we do treat already established tumors and have beneficial effects. So I predict it will work, but we'll see. So very important question.
There's just two comments, one from Sandra McAllister saying, fantastic work, perfect inaugural meeting. And then Joanna Joyce also says, thanks for a great talk and for joining to everyone for joining the discussion today. So I think it's a great idea if maybe folks want to turn on their cameras and participate in a more interactive discussion. I don't know if you want to start with this question from Adam Wright about emigrated monocytes.
Yeah, let's, I would love to hear from Lisa Cousins and maybe Enzo and others. What do you think?
Well, I had stepped out of the room to refresh my coffee.
Okay. Well, the question is, it's one that I need to know right now for publishing a paper. It's, and Adam can ask it himself because he's here. Go ahead.
Hello, thank you for allowing me to speak. And I was just asking about monocytes traversing into the lung. Traditionally, I think if monocytes came from the lung, they might have been called macrophages because they were from a tissue setting. But now with this increased diversity that we're seeing, when a macrophage looks like a monocyte, should we be calling it a lung-derived monocyte? And how do those what's the consensus among the myeloid network about naming those cells, those monocytes in the lung tissue that are in between monocytes and macrophages?
You know, Miriam Mirad's group has really done, I think, the most extensive and in-depth analysis of the lung myeloid microenvironment. Is Miriam on today? Because I think she's perhaps best versed from my point of view insofar as just detailing ontogeny and just the evolution of the cells and their plasticity.
Yeah.
Jennifer, do you know if Miriam is here today?
No, I don't see her on right now. I'm not sure if there's anyone from her group that's on.
So, you know, what I've seen is that, you know, it all depends on the markers. We mainly define cells by markers and sometimes by morphology. But, you know, if you take differentiated macrophages in vitro and you treat them with certain substances like TGF-beta, they change their morphology compared to, say, LPS-treated macrophages. So looking in the lungs, what we see is that alveolar macrophages typically look like what you might think of as a macrophage. It looks like a fried egg. But then if you look at the perivascular macrophages, and Claire Lewis, if she's still here, might comment, you know, they look more fibroblastoid, right? So they're moving, they're clearly migrating. Morphology is not,
No.
and single markers maybe are not the only way to define them. But, you know, in tissues, we use things like mouse, like F480 or IBA1 to classify something as a macrophage. But then if you do flow cytometry, you might find that there's a little bit of F480 on a monocyte. And so I also am currently wrestling with this in deciding what to call certain cells.
In the past, I was doing cytometry on human sputum samples, and we identified a population of cells which we called at the time small macrophages because they were a lot smaller than mature macrophages, and they had lower expression of scavenger receptors and higher expression of things like CD14.
Yeah.
But we called them small macrophages at the time, but as time has progressed, and I've been dwelling on, this was a long time ago, and now with single cell technologies as well, I was trying to write a review to come to the conclusion that they should be called lung monocytes. And I got myself caught up in a semantic.
Mm-hmm.
I don't know if you can hear me. This is Enzo Bronte from.
Yeah.
I wanted to point a couple of issues here. You know, one thing is the recently emigrated monocytes to tissues. And another thing is what's happening in the patients with COVID-19 and cancer, which is an altered myelopoiesis. it's clear that these two things have to be taken into account. They may not be the same thing. So you may have a number of monocytes which are not normally present during steady state hematopoiesis. So this is the first issues. But to come straight to the point you asked, in COVID-19, there is a paper just published in Immunity from Donna Farber, talking about 163 positive monocytes, which are expanded in COVID-19, and they are found to be expanded in the blood and also in the bronchoalveolar lavage. So they tried to connect these two populations, and the evidence they provide is that the monocytes in the lung are probably derived from the blood, but it's unclear whether these are recently derived or not. But this looks like a major alteration. So I was thinking when Judy was talking, whether they have a granularity to define better the 163 population, whether we really know that these are all macrophages, because we have seen some peculiar pathological aspects of alveoli filled with cells that don't look like macrophages, but they're 163 positive. So we don't know how to really call that.
Yeah.
So I don't know whether this is going to solve your issue, but of course, this is a dynamic conversation.
Yeah, it does touch on the recent literature suggesting that one of the newer dendritic cell populations, DC3s, which are CD14 bright, CD88 and 89 negative, and CD163 positive. On that confusion about, yes, as you're saying, whether they're monocytes or whether that have expressed CD163 or whether they're DC subsets.
Can I just make a comment? Does it really matter? Why we're so obsessed whether they're monocytes, macrophages? And all data from all this single cell analysis on coming to the one point, we're forgetting why we really study the cells. We study it because of function. And it just really doesn't really matter whether they're monocytes or become macrophages. We know that this is a process which takes place inevitably. And that's what we probably need to be focused on. There's a commentary actually a couple of weeks ago from Abrams, Scott Abrams, in Cancer Cell. Small commentary, but it's very actually close to my heart. He argued that there's only two types of macrophages in cancer. It was a cancer, not COVID. So-called defenders and so-called tumor promoters. And it's a simplistic view, but I think this is really what matters, not what the functionality, phenotypical characteristics, or whether they're truly monocytes or macrophages. So I think that we probably, the Ph.D. gamma does not address the question whether there will be truly monocytes or macrophages. The question is how they behave after you block it. So that's, I think, the most important thing. The rest is not important in my view.
Yeah, thanks, Dimitri. I mean, I was going to make that comment, though. I mean, it definitely, obviously matters to a lot of us how to define the cells, but ultimately...
Yeah, because you want to publish paper, that's important.
Yeah, we have to publish a paper. So people deal with that by saying monocyte / macrophage. I mean, so I'd say maybe it's not always possible to tell whether it's a recently arrived monocyte or a differentiating macrophage. But I think that it's obviously a question that's very timely and changing. So.
Yeah, but I agree with Dimitri. I mean, if we can combine the things and have a deep resolution and understanding whether really these cells have a peculiar molecular or metabolic asset that might explain some peculiar functions, that will be the target. So until then, I guess we are talking about possibly an infinite, continuum, and we are starting just to discover single events. I'm sure it will take time before we fill the gap of our knowledge in terms of differentiation of cells.
Yeah. Obviously, things that we're all struggling with for publishing. So great discussion. Thank you, everybody. Thank you so much for attending, and let us know what you think, what you'd like from the network, how we can help. And if you're looking for postdocs, please post it on our website. I can tell you it's effective. And let's keep the discussions going. I look forward to the next talk, which will be by Joanna Joyce. We look forward to it. So, thank you, everybody.